the magic of theater.
All right, folks, we're gonna get started here. Thanks for your patience. We're waiting to see who turns up with our snowy weather. All right, so a moment of quick introduction. Move to our introduction slide. I am Thomas Neufer. I am a PhD student at UMass Amherst in organismic and evolutionary biology. I'm going to be the MC for the evening, and I'll introduce our speakers in just a moment. But first, I'll tell you a little bit about Science Stories. So Science Stories is a graduate student organization run by graduate students in the natural sciences. We've been operating since 2011 to bring the science that we do and that local researchers do into our broader communities here in the Valley. And we do that by running these science cafe events, as well as the That's Life Science blog. Often we're interviewing folks who are a little later on in their scientific career, but today we have a special panel made up of our own graduate student members. So we're going to peel back the curtain. I'll just say personally, a few years ago really, I had no idea actually that graduate students were working scientists. So I hope that, and much less that we were also researchers and teachers and mentors and all these other roles that we fill. So today, hopefully, we'll provide for you a little bit of a behind-the-scenes look into what being a graduate student scientist looks like. So I want to welcome our speakers. We have Andy Doig in the chemistry department, Josh Holtzman in food sciences, and Janelle Welton in biology. And actually, I want to give each of you the chance to introduce yourselves. So we'll go through one at a time, and I'm hoping you can tell me a little bit, your name again, your lab and department, what year of study you're in, and your area of research. So maybe we can start with Andy.
Sure, yeah, so I'm Andrew Doig. I have started my third year in the chemistry department, but I'm in molecular and cellular biology. And I've been working in the Streeter group now, where we study protein degradation. We're a lot of biochemistry in that laboratory.
Hi, I'm Josh. I was up until a few weeks ago a master's student in the Department of Food Science, and I've just been accepted. I just accepted the offer to continue on as a PhD student in Matthew Moore's lab, where we're studying human norovirus, which is one of the most prevalent and one of the most taxing foodborne viruses in our food system. You'll hear a bit more about that later.
Hi everyone, I'm Janelle. I am a fifth-year PhD candidate in the molecular and cellular biology program, but my lab is actually in the vet and animal science department. I study early embryonic development, so looking at the really early stages when you are just less than a trimester old and seeing what genes are necessary for development.
Great, so today I want to get the chance to talk about your research and the broader context of your research, but before we do that, we'll want to hear a little bit more about each of you as people. So, in no particular order, would each of you tell me what you were doing right before you started your PhD at UMass?
I can go first.
Sure.
So before my PhD, I originally in undergrad knew that I wanted to get a PhD, but my senior year was so stressful and I knew I couldn't apply and put in like the effort that I needed to apply for schools. So I took a few years off and I worked at Nova Biomedical, which is a biomedical company that makes different blood gas analyzers for like hospitals. So I was in their research and development department working on building new assays for hospitals. Wow. It was really cool.
I graduated from Oberlin College in 2021, and after that I decided to take a few years off to get some more laboratory experience. So I was working at it as a lab tech for Gary Ruvkin's lab at Mass General Hospital. If that name sounds familiar, I don't want to flex on all you here, but Gary Ruvkin won the Nobel Prize a few weeks ago. So I can now say I've worked for a Nobel Prize laureate, which is pretty cool.
That's phenomenal.
Yeah, he worked with C. elegans. And you'll hear more about that later too as well.
Yeah. So.
Speak that.
Yeah, right. I had a very different trajectory. Before coming here, I was a media producer for several years, and I decided that I wanted to all that work is very ephemeral. I wanted to produce something that was going to last. And I always thought science is something that benefits people, and if you generate something that contributes to science, it can last forever, right? So I ended up going back to school, and I was looking to be a science teacher at high school level. And then I was enjoying that experience so much, I changed over to an actual biology degree, or the Bachelor of Science, applied to grad school, and now I hope to be an educator for a undergraduate-focused university. So that's why my path.
That's great, and Andy's production experience has not gone to waste. He's massively upgraded our Science Stories tech infrastructure, so if it sounds good or it looks good for folks on Zoom, or if you're watching our recordings after the event, you can thank that non-research background.
yeah, it's really been a great opportunity to use it again with Science Stories. So.
Yeah, well, we're so glad to have that experience. So you all just told me a little bit about the sort of path from being maybe a college student or, a young adult into leading your own research. So let's go back even earlier. That's what you were doing right before you started your PhD, but when you were a little kid, like 4 or 5 or 6 years old, do you remember what you wanted to be when you grew up? Janelle, embryonic development, was that on the top of your mind at that time?
I can't say that was on my mind, though I was, as a child, doing some experiments with worms that are very similar to developmental bio experiments. The little kids, when you're a little kid and it rains and there's all kinds of worms, you go collect them all, but then you learn you can cut them in half and have 2 worms because they'll both be alive. I learned that at a very young age and just kept finding out how far you could cut them, which is actually one of the most fundamental developmental bio things that is done on planarians and flatworms. That's something that they do, where they cut the flatworm at different parts to see what can regenerate and what can't.
Wow, so I hope anybody who also cut worms up as a child feels really bad that they never leveraged that into anything critically useful like Janelle has.
They shouldn't feel bad. They should know that they were already being scientists as little kids.
That's a great way to look at it. Yeah, starting early. So what about you, Andy and Josh?
Oh, sure. So my father was a scientist. He was a biochemist, became a biochemical engineer, and was consulting when I was a kid. And I thought that maybe I would do that when I was very young. In my adolescence, I rejected my father for a few years, right? And so I left the science behind, and I'm very thankful that I picked it back up. But actually, I had kind of thought I'd be a researcher when I was very little, to be honest.
Well, I was burning ants with a magnifying glass to see what would regenerate. No, I'm kidding. When I was very young, I used to love going to Boston's Museum of Science, and I was always completely enchanted with how they presented everything. And for a really short while, I thought I was going to be a museum curator. And that was me at 7 years old, not knowing that museum curation was not science communication. It was not necessarily all about science communication. You'd have to actually get an art degree. And if anybody who's watching knows me, they know I'm a terrible artist. Seriously, you ask me to draw anything, it will look like a blob, no matter what. Even if you ask me to draw a straight line. So curator, and then I was always interested in science. I never really knew what I wanted to do though.
Well, that's great. I mean, I don't know. I hope that this doesn't reflect on my commitment to my own scientific research, but I wanted to be a trapeze artist for what it's worth, and have not followed through with that particular. It might be too late for me by now, but thank you. That's very generous, Andy. So, Andy, you mentioned that your family has a scientific background. So, maybe for you this doesn't apply so much, but one of the things we wanted to hear from each of you is sort of, what's your description of your work versus, maybe what your family or your friends, maybe those outside of the sciences, think that you do? Whether big picture or in terms of the day-to-day.
Yeah, for sure. I mean, I was just at Thanksgiving dinner, right? And so how do you explain to your family what your science is something that comes up at Thanksgiving? And basically, I end up describing it in very broad terms. Well, you guys know proteins? I look at the way the proteins are degraded, and you guys have heard DNA? What's the intersection between DNA and proteins? And that's often the level that like a dinner table setting you can engage with. But at the same time, like you open doors for people who are interested, right? If they ask more probing questions, then that's actually a really great avenue to start exploring those ideas more in depth. So I don't mean to be flippant, but oftentimes you do have to give a higher level explanation of what you're doing just so that it's accessible to people. But generally what I tell people is, yeah, I'm intersecting, what I'm looking at is a protein, it's involved in protein degradation, and it also seems to be involved in DNA packaging and regulation, and I'm trying to unwind what that DNA packaging role is in this protein.
Any conversations at the Thanksgiving table about either of your work?
Mine, most of the time, my family just knows that I work with mice, and that's all that they can remember, is that I do something with mice, and that I get upset when the mice aren't pregnant. And that's what they know, which is true, because the mice being pregnant is necessary for my experiments, but that's all that they can really always remember. If we go back to like my slide, I know that I had a little like graphic of,
This one?
yeah, either one is fine. So I put the little like on-off switch because that's kind of how I describe it to my family, where I say normally when in your regular cells you have all kinds of genes and they're all on and you get a happy little mouse. But for what I do for my research is we turn off one gene, and when we turn off that one gene, we no longer get an adult mouse. And we have to figure out why. And so that's what I look into. So turning off one light switch and trying to figure out what did that light switch actually go to and control.
Right.
Yeah.
Well, can I get my meme slide?
Yeah.
Yeah, because my meme slide says it most concisely, is
There we go.
worms can get mouse diarrhea. Yeah, so over the past 2 years, it's been my task to sort of distill this down for my family and like lay people, and I eventually turned my elevator pitch into worms can get mouse diarrhea. It's a means to an end though. It's basically the lab I've been studies human norovirus, which is one of the most taxing viruses on our food system. If you've ever heard of the latest like food poisoning outbreaks on like cruise ships or gas station sushi or whatever, it's almost always human norovirus. It's very resilient. It sticks around. It's hard to disinfect. One of our most recent interesting findings was, can I get the next slide?
Sorry, just a moment here.
Was that supposedly norovirus? This is a surrogate called norovirus, or the mouse version. Can actually stick inside the worm and potentially replicate inside the worm, which is kind of interesting. And it's interesting for a few reasons. In ways, in reasons that will help us sort of study the virus and also give us a reason to make more in the lab. And so basically, worms can get mouse diarrhea, and that's sort of the start of the conversation usually. And then I usually go stepwise into like why that's interesting.
So that's a great conversation starter at the Thanksgiving table.
Exactly, yeah.
I have to say, I've been at family Thanksgiving where norovirus was present as a conversation topic and present in the room, and some of my worst holiday memories of all time.
Let me just say, I don't usually bring this up at the dinner table.
So, having gone through these sort of, and actually, I think Andy, you have some. I want to share your memes too.
Oh, yeah.
For your genetic information there.
Yeah, so I don't know, this meme is probably outdated, but back in the day on MTV, there was a thing called, it wasn't Cribs, but anyway, that era, and they would pimp out cars, Pimp My Ride. And Pimp My Ride was great, and they would do things like put speakers on speakers, because, bro, I heard you like speakers, right? And when I joined my lab, one of the reasons I joined my lab was because I had this opportunity to work on epigenetics. And the cool thing about epigenetics is that it's a way of encoding information that's outside of like the canonical way that we do that, which is like your Watson-Crick base pairing, right? Your classic DNA double helix. And it turns out there's information in the packaging also. And that idea, when I first heard about it, struck me so much that I was like, I want to learn more about the way we transfer information that's outside of like the canonical ways. And so that's why that meme is so good to me, because it's like, okay, that's what is inspiring to me. Yeah.
So outside of your sort of family or friends' perception of your work, have any of you faced any kinds of myths or misconceptions or stereotypes as scientists at this point in your career or any other point with people you know, with the general public, or even your colleagues?
I typically, like, if I start to explain an experiment, I sometimes get people kind of immediately thinking that I'm doing, like, evil things in a lab. If I talk about, like, talking about things like reporter lines, where we genetically modified a mouse to fluoresce the color red when it produces a certain protein. It's a very useful tool for us to be able to look at that protein and see where it goes. But that is a crazy thing to do, because you're putting in like a piece of a jellyfish into a mouse in order to do a thing. And like, that's a really scary kind of like, oh, you're an evil bad scientist combining organisms things if you don't know what it is and if you don't like talk about the tools. So that's like one of the things I've had to deal with a lot.
Yeah, and I want to follow up on that before we get to our other panelists' answers, because I think that, you know, working with animal subjects is something that a lot of people outside of biology don't have much hands-on exposure to and don't necessarily, you know what that looks like or how folks go about it. So you're working with live mice in the lab. Are there any kind of practices or protocols that you have to go through, you and your coworkers, to make sure that those animals are being, you know, handled in a careful or humane kind of way?
Yeah, there's a lot of protocols that we have to do. Everything that our work is doing has to be approved by, it's called IACUC. It's an institutional thing where there are both members of people that are in the UMass community, and there are at least 2 members of that board, I believe, that are outside of UMass, so they are independent, reading over these protocols to make sure that they are doing the best for the animals. All animal research has to follow like it's called the 3 R's of animal research. It's like reduce, refine, and replace. So try to reduce the number of animals that you're using at any point in time when you can. Refine all of your protocols so that you're using the most that you can from every animal, and replace whenever necessary. So if you don't have to use a live animal, can you do a different assay? So that's kind of like all sorts of guiding principles to make sure that you're doing what's best for the animals and still taking care of this was a live creature.
Yeah, great. Thanks for sharing that. What about the 2 of you, or do either of you have maybe? Yeah, Andy, do you want to start?
Yeah, I mean, I think one thing that people don't know, and we'll maybe talk about this more, is that there's so much more to being a grad student than taking classes. So like, yeah, we're researchers, but the other thing too is that like you do a lot of mentoring. So I always have an undergraduate who's working with me, and I always have, at this time of year anyway, a rotation student who I'm working with. And a lot of my time is actually spent with those students on their projects. And you know, that was not something that I expected. I essentially expected that I would be receiving all the mentorship, you know, and that inversion was very different. But this, as far as like what people think I do, like, you know, I don't wear a lab coat, but every day I am like mixing chemicals together pretty much. So I actually fit that kind of role.
I think you've got kind of a classic scientist.
Yeah, exactly.
What about you, Josh?
Well, first to build off of your animal thing, people always wonder why are you studying worms? It's like so I don't have to deal with mice. I don't have to deal with any of that. Mice have all these other regulations. But moreover, people ask me, you know, mice. Sorry, I was thinking about mice. Worms, viruses. What does this all have to do with food and food science? Because people mostly think of food science as like, oh, we're like growing meat in like a test tube. And yes, there's like a little bit of that going on in other labs. But food science is, you know, so broad and so multifaceted that really any intersection of science and food can be called food science. So really, there is a little bit for everyone, but I do sometimes have to explain that, like, you know, even though I am studying, you know, worms and viruses and stuff, it still relates to food because it is a foodborne pathogen. It still affects our food systems in a lot of sometimes very scary ways. So yeah.
Yeah, thanks for sharing that, and I think we'll have a chance in a little bit to get into some of those broader contexts of the work that each of you are doing now that we've heard the kind of quick elevator pitch. So I want to get into the day-to-day of your research, but before I get into the day-to-day of your research, because we can all be sometimes a little too work-focused, I think, as graduate students, when you're not at UMass, when you're not mixing chemicals together, or mice and jellyfish, or worms and viruses in the lab, where would we be likely to find you, and what would you be doing? If you're ever not mixing chemicals together in the lab during waking hours.
Oh, you mean like my cocktail hours?
Yeah, there you go, different kind of chemicals.
Yeah. No, I'm a huge outdoorsman and mountaineering. I try to think of my life as I have two careers, and I should pretty much be working on either one of them most of the time. And the other one is my mountaineering career. So I'm going to Alaska. I'm going to do some tough trips out there over the summertime, and so I'm trying to stay fit, and I do a lot of training for those kind of things. So if I'm not in the lab, I hope I'm outside.
Oh, cool. What about you, Josh?
Nowadays I'm trying to keep my love for singing and music alive. Back at Oberlin, if you're not familiar with Oberlin, they also have a conservatory, and I tried to involve myself as much as I could with that. And as a result, I actually got pretty far without actually being a conservatory student. And as a result, I sort of began to realize that maybe singing can be this sort of lifelong hobby. And coming to the Pioneer Valley, there's first of all we're in the 5 colleges system, which is what actually started as just the different libraries of Smith, Mount Holyoke, UMass, Amherst College, and
Mount Holyoke? Did you mention Holyoke?
Yeah, Mount Holyoke, Hampshire.
Hampshire.
Altogether. But that sort of turned into sort of this larger network of sort of collaboration, which now allows me to sing with Amherst College's choral society. I'm also singing with a local group called Illuminate Vocal Arts Ensemble. They're actually all of them are actually directed by the same person. So I actually auditioned for Illuminate first, and then she was like, "Hey, do you want to join Amherst College Choir?" I was like, "Sure, why not?" So nowadays I spend a lot of my free time rehearsing for these different groups and singing wonderful, challenging pieces. When I'm not doing that, I'm usually just vegging out on the couch.
Yeah, I know how that goes. What about you, Janelle?
Yeah, I've sat here trying to think of like any other things that I do right now. And being in a 5th year, like I am trying to defend in the spring. I'm also teaching this semester and next semester. So I have not had a lot of free time. So most of the time if I'm not in the lab or teaching, I am at home and probably asleep.
Yeah.
Or painting my nails. I got some nice Wicked nails today,
Ooh.
but I do like doing that.
That's great. I noticed that, and I wondered if that was a Wicked reference.
Have you seen it yet?
Not yet.
Okay.
I will this weekend.
No spoilers.
I won't spoil it, but I was a little bit of a
God.
tangent. I was a Wicked super fan as a child, and so I went in, I feel like, ready to be quite judgmental, but it was beautiful. I was really moved. I saw it last night, so, go see it. Not that they need me certainly doing their advertising work for them. I feel like they've postered it up on the inside of all of our eyelids. Okay, so that's what you're doing when you're not at work. When we find you in your workspace, what does your average day actually look like? What do you do? Some of you got into this a little bit, but if anybody wants to flesh that out for us a little bit.
Yeah, for sure. So I do a lot of bioinformatics work. So there's the preparation of the samples. That's where I'll actually do wet work, right? So I developed some cell lines. I needed to do some assays with them, enzymatic assays and so on, and processing them. And actually, in that photo, you can see a bunch of the buffers and so on behind me there for doing these kind of things. Doing some protein purification now. So that's all things that I would do at the bench. And then there's also just a bunch of learning how to compute these large data sets. So that's sitting in front of a computer, learning how to use all these command line tools, and then visualizing data in R. So that's the other side of things. And of course, you try to fit your reading in and your additional background where you can. A lot of presentations and public speaking for me, part of CBI. So that's the chemical biology interface. I got a traineeship through that, but there's additional course load and so on for those things. So yeah, the general work day, it varies on what state of the research I'm in, but it's usually beginning in the morning at the bench, and then afternoon try to get to the computer.
And when you say, sorry, I'm just going to ask you to define a couple of terms.
Yeah.
When you say R and when you say bench, what do you mean by each of those?
Well,
things?
yeah, so the R is like a programming language that's really effective for working with particular types of data. Easier to use that to manage really large data sets like you get out of next generation sequencing than to try to do it in Excel, for example. And then the bench is what I would call any time that I'm working with actual cells or chemicals or proteins, and
Doing like physical experiments.
doing like physical experiments, prepping the raw materials.
Yeah, there you are,
for-
Ben.
That's the bencher there.
With the
Yeah, that's the
with
bench on the
photograph
right
it,
there. Yeah.
Yeah, great. Okay, well, what about either of you?
It really does depend on what different experiments are going on, and also I actually am working in two different labs because I am working with the virus side and the worm side, and those require different procedures and or we'll call protocols. But moreover, I'm actually working in two different lab spaces for each of those. So some days I do spend a lot of time. The worm lab's on the second floor, and the virus lab's on the fourth floor. So running up and down the stairs. You do not want to use the elevators in Chenoweth Laboratory. The elevators in Chenoweth Laboratory are haunted. I don't believe in ghosts, but they are haunted. Do not use the elevators in Chenoweth. Anyway. So often it really depends what experiments are running. If I'm doing something more cell culture focused, or something more worm focused or virus focused. Right now I'm developing a workflow for just trying to get the infection going in the worm, and that requires a lot of making sure the worms are growing and preparing them for infection, and then infecting them and collecting data from that. But when all that's not happening, I usually I'm reading papers. Right now I'm working on a review, so I'm doing a lot of reading and preparing, right now I'm working on drafting the figures because I have most of my manuscript ready. But I'm not in the building all the time though, which is if you schedule your experiments where you can actually have some time and some work from home days if you're clever about it, and especially when you're waiting for things to kind of grow up and get to the right life cycle stage or whatever, and
Yeah, Janelle, you said your schedule sounds pretty packed. You mentioned teaching, you mentioned lab work.
Yeah, right now, this semester has been a very busy schedule because I've been teaching a biochemistry class and doing lab work and writing up a manuscript, and also helping my undergrad who is working on her honors thesis. So a lot of busy things, but I did have one slide that has pictures on it. That's one of my favorite things to do in the lab.
Just give me a moment here, sorry about
Yeah,
that.
sorry. One of my favorite things to do in the lab is something that will show up in a couple seconds, that actually takes about like 3 weeks to arrive at this result. So these pink and green photos that are here are immunofluorescent images, which I'll explain what that is in a second. But it takes about 3 weeks to get to this point because what those are is actually mouse embryos. So that top one is what a normal mouse embryo would look like at day 7.5, which is about a third of the way through a mouse pregnancy. So that's what it would normally look like. And then on the bottom, it's when I've turned off my gene that I care about. And so what you can see here is that I had to take these physical samples and I had to process them, I had to put them into a piece of wax and then slice that with a microtome, which is kind of like a deli slicer. So slice it like deli meat, put that onto a glass slide, and then do a 2-day protocol and finally get these images.
Wow, but at least they came out Wicked themed.
I mean, I can change the color at any point, but what these actually show you is 2 different proteins. So in pink, it's marking one protein that's being expressed in a specific tissue. In green, it's marking a different protein that's in a different tissue type. And so in the top one, you can see a little bit. You can see there's 3 distinct layers with a black space in between. And in the bottom, there's no layers. There's just all one thing. So
Which
that's
is
one of the...
bad.
one of the types of assays that we very routinely do in our lab is this immunofluorescence, just looking at different proteins every time.
Great.
Yeah.
Do either of the two of you have a recent result or a kind of result like this, finding that you're excited to see once you've finished your protocols or when you get your results back?
Yeah, for sure. I'm excited about pursuing some transcription factor binding sites. So if we take a look at the slide here down at the bottom, and unfortunately this, what they call a volcano plot, the gray does not show up on the projector, so it might be hard to see. Maybe you at home can see it better. But we have been finding that this protein I was describing earlier seems to be augmenting the access of DNA. So like you can improve the access to DNA, and things that work on the DNA to read that information will be more effective. The things that modulate that activity seem to be preferentially changed at sites called enhancers, which are places where things called transcription factors bind and regulate the amount of protein that's made off of the DNA. And the fact that I was finding more of these enhancer sites being impacted than other sites was novel and exciting, and suggested a couple of mechanisms. And really in a biochemistry lab, what you're trying to do is narrow things down from like a descriptive statement where you're looking at, oh, it seems like enhancers are being affected, okay, but you have to go into show the chemical mechanism for that happening. And so the fact that I now have something that I can isolate and hopefully regenerate in a tube, which is like the holy grail of the biochemist, is really exciting for me. Because it not only does it mean that new knowledge might exist there, but it also means my thesis is going to progress.
That is
one step closer to graduation.
That's what it comes down to, right? What about you, Josh? Anything you're excited to find recently or hope to find?
Well, the fun thing about the stuff I'm doing right now is that it's very foundational work. You know, C. elegans, the worm that I'm working with, is a very widely used biological model. So a model organism is like an organism that can study a much more complicated thing in a much simpler way. So for example, people here have been talking about cell lines and mice. Those are all different model systems or model organisms that you can take a very complicated thing and make it very simple, or well, relatively simple, let's say. Because to be honest, it's not always simple, and in this case, C. elegans has been developed as a model for everything from like neuroscience to biochemistry and development. One of the things it hasn't really been developed as is a microbial model, dealing with tiny little organisms, and specifically, it really has not been robustly developed as a viral model. In fact, up until about 2011, we didn't know of any natural viruses that infected worm. Back in 2011, the Orsay virus was discovered on a worm feeding on a rotting apple in an orchard in Orsay, France. Which, if that isn't the most French thing you've ever heard today, then I don't know what is. But that also makes what we're finding a lot more remarkable, which is that we're now seeing that another virus can naturally infect C. elegans. So what I'm doing right now is just trying to get that to happen consistently and get replication, get replicable results. So that's also another big thing about science, that you have to do the same thing multiple times to make sure you have enough data to get significant results that you can publish and say this is happening without a doubt. So that's what I'm doing. I'm trying to get consistent results, trying to get consistent data that is consistent with previous people's work as well.
So having talked a little bit, and we'll come back to some of your research, but I want to pivot and think about kind of grad student life. So one question that I have for each of you, and also I just want to, I'm going to interrupt and just say for a moment that if anybody who's attending on Zoom has any questions, or anybody in the audience has any questions, we're a small group today, so just raise your hand, or if you have a question on Zoom, pop it in the chat or in the Q&A, and we'll read it out loud to our panelists. So we'll have a chance at the end to ask questions as well, but don't be afraid to jump in. So I want to ask each of you how you chose the lab that you ended up in and how you chose UMass. Did the lab come first? Did you decide you wanted to be at UMass? Did you just pick out of a hat?
I decided UMass first.
Okay.
I was between UMass and University of Vermont. That's the other place that I interviewed at and toured and everything. For me, I chose UMass because it was out of my comfort zone.
In what way?
So I came from a small town in Maine and then went to a small college in Boston. So like most of my class sizes for all of my life have been like 20 people or less. So coming to UMass and like seeing how big everything was really alarming. I also started my PhD in the fall of 2020, so weird time to be starting a PhD. So that factored into my decision of like going to Vermont or staying in Massachusetts because of family and location as well. But once I was here, I chose the lab slightly by accident, rotating in it. I actually wanted to rotate in our partner lab, which I did, but I couldn't rotate it in that lab for my first one, so I just happened to choose this one and do it as well, and turned out I loved it.
So Andy mentioned rotations as well. Janelle, can you give us a quick and dirty what's rotations?
Yeah, when you become a PhD student, when you're first year, you don't know what lab you're going into, so you try on a few different labs. So for like 6 weeks, you are thrown into a lab, you get given a project, trained up, do some things, and it's a trial period for the PI to see if they like you and like how you work, and it's a trial period for you to see if you like the PI and you like how the lab functions.
Oh, that's
And you like
interesting.
their like mentoring style as well, and like
Yeah.
the atmosphere of the lab.
Yeah, so you get the chance to check it out. Yeah, that's
Yeah.
interesting. We don't do that in ecology. We don't have rotations to
Oh,
apply
yeah.
directly into a lab. So
Yeah, so I got to rotate in a few different labs, working one was with zebrafish, 2 of them were in mice, and one of them was more similar to my undergrad research, working with like biophysics and doing microscope things. And I chose to go outside of my comfort a little bit to learn something new.
yeah, I came from a small liberal arts college, and UMass felt like such a huge, huge transition, big step out of my comfort zone too.
Yeah, but I didn't want to deny myself the core facilities and all of the resources that UMass has just because I'm intimidated by it.
Yeah, it's great to be somewhere that has those resources, like to support the research that you want to do. So what about each of you? Why did you choose, Josh, why did you choose UMass? And choose to stick around for a PhD as well, so you must be liking it okay.
So yeah, I was initially looking for a master's program in food science because food science has been sort of this lifelong interest of mine. I was also simultaneously at the same time applying to PhD programs to go directly into molecular and cell biology, something closer to you guys. But at the same time, I was interested in exploring this interest of mine, which I had developed in high school and college, and actually developed a course and taught it back at Oberlin for 5 consecutive semesters, which I'm very proud of. But it also made me realize maybe I want to do this as a research career. And I looked at various programs, and one of the great things about UMass is that UMass Amherst has the #1 food science program in the country. It's #3 in the world, which is pretty great. And I also already had a connection with Dr. David Julian Clements, who has his own lab here as well. He does something I'm not well versed in, which is sort of materials science kind of materials chemistry as well. But when I was looking at different projects, when you're applying to a master's program, you also do something called direct admit, which is where you don't rotate. You have to find a lab. You have to reach out to the person who's running the lab, the PI. What does PI stand for? Principal investigator. No, I had personal investigator in my mind. I was like,
Private eye.
Private eye, yes. But the PI or the advisor or whoever you call, whoever runs the lab, you have to reach out to them, talk to them about any open projects they may be able to take, put you on, and if they have room for you in the lab. So if you're thinking about doing a master's, especially like a research or a thesis focused master's, you need to find someone who has room in your lab. And you have to make sure that the projects that they have open are ones that you're interested in. And it just so happened that when I was working at MGH, I was working with C. elegans, the tiny little worm I was talking about. And when I contacted Matt, he was just thinking about this project that they just started, where they had infected the worm with norovirus and found some exciting results. And we had a conversation about it, and I brought some of my own. I don't want to call it expertise because I'm definitely not an expert, but sort of my own perspectives from working with the worm, which Matt didn't have as a non-worm person. We call ourselves worm people, okay? And it just so happened to be a very perfect master's project, and a good thing to look for in like sort of a master's project is something that you can jump right into and get some clean results in the first couple of years of research, because for most master's projects you only have about a year of research to do.
Yeah, that's a great point about finding a project and thinking about the scope of what kind of project you can complete during the span of your degree. Andy, did you come in with a project? Did that?
I did not
So you're
come in
allowed?
with a project,
Yeah.
no. And that rotation period, which is not, I think, universal, there's a direct admit process, was one that I was relying on heavily to help find a lab. And one of the things that, at least at UMass, that's done for admitted graduate students is that at the beginning of your time, you're teaching, you're taking some classes, you're coming up to speed with what academic life looks like, and you're also receiving presentations from PIs who are recruiting for their laboratory. And so during one of those talks, my current PI, Eric gave a talk about the ubiquitin code, which was a way of encoding information using proteins, not DNA and so on, right? And I thought, well, that aligns with my interests. So we had a conversation after that, and after a few rotations, it's the lab I ended up in. So I happened to find someone with similar research interests to my own during the rotation process. And that's how, he was also an extreme athlete. So there was a personal dynamic that's so important to have that kind of relationship. So that's how that decision was made.
Yeah, that's such a great point. And I think we've got a question from the chat. You want to read that out, Matt?
Yes, we're just trying to. Yes, we have a question in the chat from Zachary. It says, "Hi, I think one of our greatest fears is failure. Has there been a time in each of your PhD journeys where you failed or had some challenge that you could not fully complete? If so, what was it, and what resources did you use to overcome it?"
One time, more than one time. Any failures to share?
So many times. But this also gets at something that I feel really strongly about, which is sharing your failures. We have like weekly lab meetings, and like usually once or twice a semester we'll present to people about our research. And I try to make it a point to every lab meeting include a slide that has my science fails on it, because they happen to all of us, and like you need to be able to be resilient, and you need to be able to be like, well, this failed because of this, so hopefully you don't make my mistake. I've told my PI I'm gonna make every mistake there is to make, but I won't make the same one twice.
I love that. Yeah, exhaust the list of possible mistakes. That's how we learn,
We'll
right?
figure out every way how to not do that
That's
assay.
the scientific method.
Yeah.
Well, yeah. The nature of science is failure. In fact, we sometimes learn more from failures than we do from successes. And to expand on what you said, a previous choir director, actually back at Oberlin, said a true professional never makes the same mistake twice, which means that it's okay to make mistakes, just not the same one twice, unless you're learning. And we're still called students for a reason. So even if you do make the same mistake twice, you are still learning from those mistakes. And to be completely transparent about it is also really important, because if you're suffering in silence, then you're denying yourself from other people's resources as well, which can be really helpful, and you can get different perspectives. Maybe if you're stuck on a problem, you might find a way around from somebody else.
Yeah, it's so true that it's often, because there might be a level of embarrassment, right? You take personal responsibility for your mistakes, right? You ought to do that because we're adults. But at the same time, it can be emotionally difficult to do that. But oftentimes the solution isn't within you, and just beating your head against the wall doesn't make it suddenly appear. And relying on other people's wisdom is the way to solve those problems. And that's a really valuable lesson to learn, and a difficult one. I think, I remember when I first joined the lab, I needed to make a cell line, and the first attempts that I tried to do that were were difficult. I tried a different method. I had a cell line that was working, and then that whole cell line seemed to stop producing the protein that I needed. And it was like 6 months of work. It no longer works. I can grow it up and it produces what's called a truncated form of the protein, which is like just a portion of what you need it to produce. And that was a serious, like, oh no moment. And then 3 months later, I had a cell line that worked and it stably worked. And the thing was to try a different construct and try it again, just essentially try it again in a different way. And then it worked. So you kind of iterate your process and adapt from the failures and keep going. And eventually something will work. You just have to find out what it is.
Yeah, I love what you said. Sorry, Janelle, do you have something to add?
I have one other
Yeah.
thing to add is one thing about like being able to be open about your failures is in finding that lab that you feel safe and have trust with your PI. I personally am in a lab where all of the grad students feel very like trusting of our PI, and like the second any problems are happening, we will go and tell him, be like, hey, this is going wrong. And like, even if it's a stupid mistake, like I set the temperature on this wrong and everything failed, I will still feel comfortable and able to tell him that, versus I've seen people in other labs sometimes who are a little bit more afraid of their PI and they won't tell them when those things are happening, which then kind of leads the grad students to not hide things, but try to work, like suffer in silence, try to figure it out all on their own instead of using your PI as a resource. So really making sure you have that trust and that two-way communication is, I think, key.
Yeah, you know, at the end of the day, like we're all workers and we deserve like a supportive
Yeah.
workplace, and we want to feel like we're in community and supported by the folks that we're working with. So I think that's such a great point. I mean, we make tons of money, so that helps, you know. But no, I'm obviously joking.
I think it's also really important to acknowledge where you are in your learning process, because if you feel like you're going to get yelled at, or someone's going to be like mad at you for some reason because of your mistakes, it's just really important to acknowledge that you are a student in science. You are at like this point in your career, and that frankly at this point in your career, it's not right for someone to be mad at you for making a singular mistake. And really anything short of setting the lab on fire should be, you know, acceptable. I mean, still things you have to learn from, and still things you have to get around. But it is so important to just acknowledge where you are in your scientific career, which is to say you are not an expert. You are not a god. You're not an omnipotent being who can see around every issue.
And something, you know, that I think is really interesting been for me about the graduate student experience is that as you progress, you know, in some ways, like you're the person who knows the most about your work. And so as much as you're aware of your own knowledge gaps, you also are moving along that. You may know more about the details of your work even than your advisor. And I feel like we just heard a lot of really wise, really inspiring thoughts about failure. So I'm going to share my really embarrassing failure, which was that I spent like 2 days debugging code I had written, and it was working. I was doing like a spatial statistics thing. It was working everywhere except Massachusetts, which made no sense. And I was combing through, you know, checking for typos, checking for issues in the code, checking for issues in the data. Spent like 2 days doing this before I realized that I had spelled Massachusetts wrong. Not just once in the code where I would have noticed it. And even I read through to make sure that there wasn't a typo. I had spelled Massachusetts wrong every single time I had typed Massachusetts.
Hey, at least you were consistent.
Exactly.
Wrong.
Yeah. I think I know how to spell Massachusetts now, but we're coming close to the end here, so I'm just going to ask you guys a couple really rapid-fire questions. We'll have a chance for any more questions from the audience, and then we'll close out and eat some pizza. So let's do these ones super fast, move through, you know, just in a couple words. What was the biggest surprise for each of you about grad school, being a grad student?
The variety of responsibilities, for sure.
The emphasis that my PI took on work-life balance. That was something I didn't really get in my previous positions. And that was
That it wasn't as straightforward as regular school is. Like, I thought I was just gonna be like, you take some classes and then you get your PhD. Turns out no.
Yeah, very different. Yeah. Okay, what about career? Like, what can you do with a PhD? Career options, goals? Where do you think you might go?
Current plans, I'm with Andy actually trying to be a professor at a primarily undergraduate focused institution. That's like my long-term goals.
Yeah, lots of teaching. Josh, you have any thoughts about that yet? I know you're just starting your PhD.
I would also agree. I think being a professor would be a great balance of teaching and research, which have been two passions of mine. But you can do a lot with a PhD. You can go into research and development. You can do like sort of government work. You could, especially in an applied department like food science. There's several options as to where you can kind of go. I'm still thinking mainly about academia. So we have academia versus industry versus government. These are sort of the three pillars of like kind of where you can go with higher education after higher education. Academia is like where you can do research funded by either government organizations or by institutions. Industry is where you can do research funded by private institutions, and government is where you do research under the guidance of a government institution.
Yeah, Andy, you said primarily undergraduate teaching is what you're interested in.
Yeah.
So that'd be academia.
That would be academia.
I do
Okay.
have one other
Yeah.
thing that I really want to. I couldn't apply this year, but next year I'm going to be applying for the AAAS Science Technology Policy Fellowship, which is a fellowship that's in DC working with senators and in the legislative branch and even the executive branch on science policy. So you write up the memos, you're the ones that are providing the research to your senators about anything that they're voting on. So that's what I'm hoping to get into as well.
Yeah, huge impacts from that kind of work. But if you were not going to do any of these things, if you were going to leave your field of science and do a different career entirely or a different scientific discipline, what would it be? You could be a trapeze artist.
I want a hobby haberdashery. I like calling it that. I call it a hobby haberdashery because, which is just, haberdashery is like a button and I don't know, pin shop. But I want a hobby place where like you bring in, I don't know, colored pencils, markers, all of the things that you need for different creative hobbies that are expensive, and you probably are gonna, if you have ADHD, buy all the things and then stop doing that hobby. So instead, you could go to my hobby place and you could come in and rent some time here and use all of the supplies.
Wow, I love that. I don't want to wish against your career and important policy, but I hope that you open a hobby haberdashery.
I hope to one day.
Happy haberdashery so that I can go do some of that. What about you, Andy? What are you thinking?
I mean, I love to travel, and I love to do it in sort of like inexpensive, adventure-y way. Like I like to live out of my van for extended periods of time, and I thought for a little bit of time that I would be some sort of great essayist in writing impactful philosophical things, and that never happened.
There's still time.
But for that one, maybe there is still time. So if I get a chance to explore that more in my old age, perhaps I would love to make it happen.
What about you, Josh?
There's a running joke in the PhD community when things get tough. Sometimes PhD students say, "I'm seriously considering dropping out and becoming a baker, and just opening my own bakery. This would be easier than doing my experiments right now." And that's something I've considered as also coming from a food science microbiology background. Something that just randomly popped into my mind literally 2 minutes ago was, which is going to be completely out of left field for literally everyone that knows me, is a truffle farmer. Because actually, in a previous life, I probably would have been like a mycologist, someone who studies fungi. I don't know what I would do without science. Maybe music? I don't know.
There you go, your choral career. I have to say, I've known some bakers, and waking up at 3 o'clock in the morning and throwing around 50-pound bags of flour sounds like one of the few things more exhausting than getting a PhD to me, but certainly I'm glad they're out there. All right, so we're at time here, so I'm going to wrap us up. If there are any last audience questions, yeah, go ahead. Do you want to come up to the microphone?
Okay, well, thank you guys so much for sharing your experiences. One question I had for you through all of this, long hours, I know nothing about getting a PhD, so, what keeps you going? And you can tell me, even if it's one sentence each, just like when you're really in the deep hole of things not working or failures repeating themselves, what is it that keeps you going through all of it to the end of the PhD and beyond?
Yeah, so I love climbing mountains, and I just think of it as a mountain. And oftentimes when you're climbing mountains, it's extremely taxing and also terribly boring.
Sounds like a lot of fun, Andy.
Well, but
Bring me with you next time.
No, that's really it. Like, how
Really
is,
selling it.
yeah, selling it, right? But like, how is something like that when you're going through in the moment that's so terrible, like rewarding at the end? Why do you do it? Because when you get to the top, the views are fantastic, right? And so I think, if you focus on the goal, my mother would say, keep your eye on the prize, then you can get through a lot.
For me, that prize would not necessarily be the degree at the end of it, but rather the big questions. And one of the fun things about being in microbiology is that there are a lot of big questions, and a lot of really fantastic and interesting big questions that you can think about forever, and just kind of admire sort of the scale at which this is all being done. It's also comforting to know there are other scientists, like usually just 100 feet away, doing also trudging through the mines and through the trenches, I guess. And I also sometimes when I'm holding up my pen, I'm thinking, wow, look how far I've come that I'm just holding this so confidently. You have to focus on sort of little things too, like little skills that you've acquired. Like, oh yeah, I am a scientist.
Yeah, recognizing our accomplishments, that's a great thing. Janelle, you're so close to the end. What's going to get you over the finish line?
You see, that's the funny thing. You guys gave like such nice uplifting kinds of
Oh,
answers,
no.
and like,
Oh, no.
don't get me wrong, like there's plenty of those where I
We got
think about
nihilist
like
here.
what I want to do in my future and be like, hey, you need the PhD in order to do these things. Sure. But in reality, what gets me through is that I would be too embarrassed to have to explain to all of my family why I stopped now. Like, I'm too close. I've already started, I've already gotten this far in, and also my oldest brother is now starting to apply for PhD programs, so I gotta be the first doctor in the family.
Right.
At this point, I gotta beat him.
You got something to prove.
So, yeah, so it's not the best way to get through, but like,
Whatever it
sometimes,
takes.
like, it would be more embarrassing to have to explain than the pain of just like my experiments not working. I can suck that up.
I love that as a point to end. So thanks to each of you so much for, oops. Well, just lost my, hold on, I can pull this up again.
I'm making that my profile.
Yeah, that's a great
I'm
last.
making that my profile picture.
Yeah, right. It's like we're all feeling sort of like Mr. Duck
I
there.
think I can pull this up again. I was just about to pull up our, there we go. So, sorry about that. Just about to pull up our closing slide here. I wanted to thank our panelists and give a round of applause and all, but also to thank our audience for showing up. Everybody, please stay tuned for our next event. That's going to happen after the new year. So you can keep up with us on our social media, which there's some flyers of around the room. You can also find that on our website, which we'll link in the chat at scistories.org. We have an email list that you can sign up there to hear about upcoming events, and we can also, we'll send out our blog posts from our graduate student scientist blog. So if you are a graduate student scientist and you want to write about your research or other science topics you're excited about, please get in touch and you can write a post for our blog. Also on our website you can see videos and transcripts of our former events, and eventually a video and transcript of this event. And then the last thing that I want to ask is that if you get the chance to please fill out our post-event survey, there's a QR code that's going to be on the screen, and we'll link it in the chat as well, and that will help us figure out anything that we can change or improve, or what kind of topics you'd like us to see it cover in future science cafes. So thank you everybody so much for being here. Thank you Andy, Josh, and Janelle. You've been great panelists. Please join me in giving them a round of applause, and then we'll break. Thanks everyone.